Hello everyone, welcome to my simple zoology class. In today's video, we'll be discussing on the one of the important topic, that is concentration of filtrate, also known as countercurrent mechanism. That is from the excretory products and elimination chapter. Countercurrent is nothing but what? Something which is going to move in opposite direction. That itself is called as what? Countercurrent mechanism. Now, mammals have got the ability to produce what type of urine? Concentrated urine. For the production of the concentrated urine, you require, or those two which are going to help or play a significant role in the production of concentrated urine is loop of Henle, and the other one is vasa recta. Okay, now what exactly is this vasa recta? If you remember the structure of our Bowman's capsule, that is the renal tubule, you'll remember how does this go. It's a superficial picture for you to understand. If this cup-shaped Bowman's capsule has got a tuft of capillaries, those are called as what? Glomerulus, having afferent and efferent arteriole. Now this efferent arteriole itself is going to emerge out from whom? From the glomerulus, to form a fine capillary network, okay, around the renal tubule. I'll show it in the diagram. What is this happening? This efferent arteriole is going to emerge out from the glomerulus, and they are going to form a fine capillary network around the renal tubule. Now, from this capillary network itself, there is a minute vessel emerging out. Okay, that minute vessel, what are they going to do? I'll show it in a different color. So minute vessel, I'm showing it in a green color. It is going to emerge out, run parallel to the loop of Henle. So this is our loop of Henle, right? So this portion over here is our loop of Henle. It is going to run parallel over here. So that thing which is going to run parallel over there, that minute vessel which is formed over there, that itself is called as what? Vasa recta. Please do remember. Well, how did it come out? First of all, efferent arteriole, they are going to form a fine capillary network. And there is a name also to remember. What is the name of that? That is the peritubular capillaries. Okay, I'll repeat the whole thing once again. Efferent arteriole is going to emerge out from the glomerulus to form a fine capillary network, and the name of that network is peritubular capillaries. From this peritubular capillaries itself, a minute vessel is going to emerge out, and the name of that minute vessel which is going to run parallel along the loop of Henle, this is our loop of Henle, right? So along that loop of Henle which is going to run parallel, that itself is called as what? Vasa recta. I hope I was crystal clear with what exactly is vasa recta. Now, further moving to the next continuation, that is, this filtrate is going to flow in the two limbs of loop of Henle. I'll just take the diagram directly and make you understand with the help of diagram itself. So this is our Bowman's capsule, okay, then comes our loop of Henle, right, and then our CD ultimately. Here separately I'll take the our vasa recta also, so that I can explain then and there itself without just confusion. And I'll be starting over here with 300 milliosmoles per liter, then coming to 600, then 900 and 1200. Okay, now what exactly happens over here, that you need to understand. Okay, the flow of filtrate in the two limbs. Now this is our descending limb of loop of Henle, and this is our ascending limb of loop of Henle. Whereas for vasa recta, it is quite opposite. This will be our ascending limb, this will be our descending limb. So please do remember, while I'll be explaining, pay concentration. Okay, the flow of filtrate in the two limbs of loop of Henle is going to take place in opposite direction. So here, where the DL is there, on the other hand, on the vasa recta side it is AL, where AL on the loop of Henle is there, DL on the vasa recta will be there. Okay, now The interstitial fluid. What exactly is this interstitial? Nothing but something called as tissue fluid. Okay, you can also simply understand it as tissue fluid. Okay, tissue fluid. Now, this is our cortex region. This is our inner medulla region, and finally, sorry, this comes our outer medulla. I'm sorry for that. This is outer medulla region, and then finally lastly comes the inner medulla region. Okay, now what exactly happens over here? The interstitial fluid of the kidney, I've already mentioned, that is the tissue fluid. The tissue fluid of the kidney is going to increase the osmolarity starting from 300 milliosmol per liter to around 1200. That means it is going to increase to about 4 times from the center to the inner medulla. So here, for this increase in the osmolarity from 300 to 1200 milliosmol per liter is mainly caused by our very own sodium chloride and urea. Please do remember this, that is sodium chloride and urea. Now what exactly happens over here? Actual story begins. Okay, sodium chloride. What is happening? Sodium chloride is transported by the ascending limb. Now this is our loop of Henle. So here the sodium chloride is being transported out. Okay, sodium chloride is transported by the ascending limb of loop of Henle, which is going to get exchanged with the descending limb of vasa recta. I'll write down those points in the next sheet, need not have to worry. This is our vasa recta, no confusion please. And this is our loop of Henle. Okay, so what is happening? First point, what did I say? Sodium chloride is transported by the ascending limb of loop of Henle. Okay, and it is going to get exchanged with the descending limb of the vasa recta. Then finally what happens? Sodium chloride from the descending limb of vasa recta will be returned back to the tissue region, that is interstitium. So from here the sodium chloride is transporting, going to the ascending limb of ascending limb of vasa recta, and here it is sent out to the interstitium. This itself is the interstitium region here. The line which I'm making, that itself is the interstitium region. I'll repeat the whole statement once again. Sodium chloride is transported by the ascending limb of loop of Henle over here, is getting exchanged with the descending limb of vasa recta. Again, sodium chloride is going back to its original place, that is the interstitium. Here itself is the interstitium through ascending limb of vasa recta. That is the first solute that is going to do the work, that is our sodium chloride. Coming to the second solute, that is urea. Now what is this urea is going to do? Urea here is added to the interstitial fluid itself. How is it going to come out? Through this collecting tubule. So here the urea is going to come out from the collecting tubule is also fine, or collecting duct. Urea which is present in the collecting duct will be whatever percentage or how much ever percentage of the urea which is present in the collecting duct, ultimately it will be excreted out. Later on what happens? Urea is going to re-enter into thin segment of the loop of Henle by diffusion ultimately. So it's like the repeat procedure which is going to happen. Urea first, what is happening? It is added to the interstitial fluid in small amounts. Okay, which is diffused out of collecting duct. Whatever, now for example, out of 50%, 45% has gone out. Eventually, remaining 5% also from this collecting duct itself will be taken out. Later on, what happens is urea re-enters the ascending limb of loop of Henle by diffusion. Ascending limb over here, it is going to enter over there into loop of Henle by diffusion. This arrangement of this loop of Henle and vasa recta itself is called as what? Counter current mechanism. And One more important point you have to remember. Now, the filtrate, what happens, it is going to come down. If you can see in the diagram or in the pictures if you have in your textbook, water molecules are excreted out. Now, why is the water molecules from the outer medulla to inner medulla excreted out is because you need to increase the osmolarity. Okay, you need to make sure that the filtrate is hypertonic in nature. Okay, more and more water from the collecting duct is taken out. So by what mechanism? By osmosis. So because of which the filtrate will become hypertonic in blood. That is when the kidney is going to produce concentrated urine that is 4 times higher than the initial filtrate that is found. That is from 300 it is going to increase to 1200. I'll repeat the whole procedure once again by writing down in the form of points. Please do remember. That is, what did I first start with? Sodium chloride is transported from the ascending limb of loop of Henle, correct? From the ascending limb of loop of Henle, it is going to get exchanged with the descending limb of vasa recta. Later on, what is happening? The sodium chloride is transported back to the interstitium. How is it going back to the interstitium? Is by ascending limb of vasa recta. By ascending limb of vasa recta. Similarly, urea initially it is added to. Where is it added to? Urea is added to interstitial fluid. Interstitial fluid, small amount, okay, by collecting duct CD or collecting tubule. Eventually, whatever the remaining urea is there from the collecting duct itself is being taken out. Then later on, this urea is going to re-enter where? Into the ascending limb of loop of Henle by diffusion. Questions like these have also come. How is it going to re-enter? Through what mechanism? And the answer is diffusion. Now, what happens ultimately? Filtrate, if it is going down, if you see, look at the previous page, filtrate further starting from initial one, that is 300 milliosmol per liter. So initial filtrate, further what happens? It starts flowing down from the collecting duct. That's when you get to see these H2O has been taken out. Okay. So as the filtrate flow down in the collecting duct, more and more water from this tubule will be taken off. Now how is it taken off? By a mechanism that is called as osmosis. That also I'll be mentioning over here. What is happening as the filtrate flow down, more and more water molecules will be eliminated. Water molecules will be eliminated, and ultimately what is going to happen? First of all, by what mechanism? That also you have to remember, that is by osmosis. When it is taken out from the collecting tubule by osmosis, the filtrate is going to be hypertonic. Please do remember, hypertonic to blood. That's when you can expect, please do remember this, hypertonic. That's when you can expect that human kidney is going to produce totally 4 times of concentrated urine than the initial filtrate which has been formed. So initially it was 300 milliosmol per liter. Later on it increased to 4 times to give rise to how much? 1200 milliosmol per liter. Okay, I hope I was clear with this counter current mechanism. If you have liked my video, please like, share and subscribe. Thank you students. Have a good day.